Okay, let's unpack this. Imagine starting life as just a tiny, whitish, sausage-shaped speck. A honeybee egg. Doesn't look like much, does it? But inside that single speck, well, there's this blueprint for an incredible transformation. And this isn't just, growing from a grub into a bee. It's about potentially becoming two totally different beings. If you're a female egg, you could end up as a fertile queen, the single reproductive engine of the whole colony. Or you could be a sterile worker, one of millions doing all the building, the foraging, the caretaking.
It really is one of the most profound examples of how the environment can shape development from the exact same genetic starting point.
Absolutely. And that potential, that incredible fork in the road, that's our mission today. We've gathered a whole load of source material on honeybee development, right from that first egg through the larva, the pupa, all the way to the adult bee emerging. And we're going to distill this research, pull out the really key insights, the surprising bits, and help you, our listener, get a kind of shortcut to understanding the deep, complex biology that orchestrates this whole amazing journey.
Yeah, we'll explore not just the physical changes you can see, but the hidden biological gears turning underneath, like how sex is determined, which is pretty unique in bees compared to many animals, and the fascinating genetics, the interactions, and yes, we'll definitely dive deep into how one single female egg can follow one of two completely different life paths.
We'll even look at how honeybees, they're not a typical lab model organism like the fruit fly, but they show us things about fundamental biology that we just wouldn't learn otherwise. So let's start right at the very beginning, the egg itself.
Right at the source. Like you said, they're small, usually whitish or sort of translucent, and they've got that characteristic sausage shape. But the most fundamental split, the thing that sets the stage for everything else, happens right when the egg is fertilized. Or not.
And this is where we get into haplodiploidy in bees, right?
Exactly. It's a sex determination system quite different from ours, Fertilized eggs, meaning they got genes from both the queen and a male drone, they develop into females. Diploid, two sets of chromosomes. Unfertilized eggs, just containing the mother's genes, they develop into males, the drones. They're haploid, just one set of chromosomes.
So, in a way, the queen is actually controlling the sex of each egg she lays.
Precisely. As the egg travels down her reproductive tract, she can basically decide whether to release sperm from her spermatheca. That's the organ where she stores sperm from her mating flights. And that decision, it's often linked to the type of cell she's laying in. She checks it out, probably measures it with her forelegs, small hexagonal cells. That signals her to fertilize the egg. It'll be a worker female. Larger cells, like in drone comb, she lays an unfertilized egg for a male drone. And those special queen cells, the big peanut-shaped ones hanging down, they're usually built just before they need a new queen, and she'll always lay a fertilized egg in those. A healthy queen is remarkably precise, just one egg per cell glued upright right at the bottom.
But what about worker bees? Do they ever lay eggs? I thought only the queen did that.
They do sometimes, especially if a colony loses its queen. In that situation, some workers' ovaries can develop a bit, and they start laying unfertilized eggs, which of course only become drones. In a healthy colony with a queen laying properly, if a worker lays an egg, it usually gets eaten pretty quickly by other workers. It's part of colony policing. You can often tell them apart, too. Worker-laid eggs are usually smaller, maybe a bit misshapen, and often laid on the side of the cell wall, not neatly upright like the queens. They just can't reach the bottom properly.
Okay, so egg laid, sex determined by fertilization. What's actually happening inside that tiny egg during those 3 days before it hatches?
Oh, it's incredibly complex biology packed into that tiny space. Like many insect eggs, it starts with these incomplete cell divisions. You get what's called a multinucleate syncytium.
So one big cell with lots of nuclei floating around.
Exactly. Then, different regions within this big cell start expressing different genes. That begins the process of regionalization, setting up the basic body plan, like where the head end and tail end will be. After that comes cellularization. Cell membranes form around each nucleus, turning that syncytial mass into a proper multicellular structure. And finally, gastrulation happens, which forms the basic tissue layers, like the gut.
And then hatching. But you said it's not like a chick cracking out of an egg.
No, not at all. It's quite different from how most insects hatch, where the larva has to physically break free. In bees, over those 3 days, the eggshell, the chorion, it actually slowly disintegrates. It just kind of dissolves away. So the tiny C-shaped larva finds itself already floating in a little pool of brood food that the nurse bees have already placed in the cell. It's a much gentler emergence, straight into its first meal.
Wow, dissolving its way into lunch. That's quite a visual. Okay, so the egg stage sets the basic structure, sex, first developmental steps. What about the next big phase, embryogenesis?
All right, embryogenesis. This is the process where that relatively simple ball of cells transforms into a structured, multicellular organism with a clear head, body segments, and tail region. Now, when scientists study insect development, the go-to model is usually the fruit fly, Drosophila melanogaster. Its early development is incredibly well mapped out, almost like a standard instruction manual.
The classic blueprint for building an insect.
In many ways, yeah. In flies, it's a beautiful cascade. First, even before fertilization, the mother deposits molecules, like messenger RNAs, unevenly in the egg. These create concentration gradients.
Little chemical signals saying, this end is front, this end is back.
Exactly. They set up the main axes, anterior, posterior, and dorsal ventral. Then, specific genes called gap genes read these gradients and define broad regions head, thorax, abdomen. Next, pair rule genes divide the embryo into repeating stripes. And finally, segmentation genes use all that prior information to outline each individual larval segment. And very early on, the cells destined to become eggs or sperm, the germline, are set aside as distinct pole cells.
And the initial thinking was, okay, bees probably just follow this same fly plan.
That was the reasonable assumption, especially when the bee genome was first sequenced. People expected to find the same key developmental genes doing similar jobs. And well, for many large gene families, there is strong conservation. The genes are there. They look similar. But here's where studying bees gets really interesting. The genes that act earliest, the ones setting up those very first patterns and axes, they show less conservation than expected, and maybe even more surprising. Even when a gene is conserved, its actual function, what it does in the bee embryo, might be different compared to the fly.
Okay, can you give us a concrete example of that, like a specific gen difference?
Absolutely. The classic example is a gene called bicoid. In fruit flies, bicoid is like the master architect for the head. It's absolutely essential for establishing the anterior or front end of the embryo, foundational. But bicoid is completely missing from the honeybee genome, and actually it's missing from many other insect groups too. Yeah, this was a big puzzle. If you think of these genes like essential support beams in a house, finding a major one just gone is pretty surprising.
So how do bees build a head without this crucial bicoid gene? Did something else take over?
That's exactly the question researchers tackled. They looked at the bees' version of those gap genes, genes like cripple, giant, and caudal. They found these genes largely do the same job as in flies, mapping out broad body regions. But their expression patterns in bees are actually more extensive. They cover more ground, so to speak. And crucially, giant and caudal are also provided by the mother in the bee egg, which isn't the case in flies. So the current thinking, based on the research, is that these gap genes, especially giant and caudal, have likely taken on the job of the missing bicoid. They do that early anterior patterning in addition to their other roles.
Ah, so it's like the developmental toolkit got rearranged, the function is still there, but different players are doing the work.
Precisely. It's like an orchestra missing its lead violin, but the violas and cellos step up and expand their parts to cover the melody. The music still gets played.
That's a great analogy. Any other examples like that?
Yeah, another one is the gene tailless. It defines the posterior, the tail end. In flies, its expression is switched on by a specific signaling pathway called torso. But guess what? That entire torso pathway seems to be missing in bees. Yet tailless is still expressed correctly at the tail end, and its function seems to be the same. How it gets controlled in bees without that pathway? Well, that's still unclear. Maybe it's regulated differently, perhaps after the gene is initially transcribed, controlling the protein level instead.
So the overall picture for insect development isn't one size fits all. It's more like a mosaic.
That's absolutely the key takeaway. You've got conserved building blocks, yes, but also parts that have been swapped out, functions taken over by different genes, and maybe even entirely new ways of achieving the same developmental outcome.
You just can't assume the Drosophila model applies perfectly everywhere. Studying bees really highlights this incredible diversity.
It really does. It shows us both the universal principles and the unique novel adaptations out there.
Speaking of fundamental differences, you mentioned earlier that things related to sex often differ quite a bit from the standard models. What's unique about sex determination and how germ cells, the future eggs and sperm, form in bees beyond just the haplodiploidy?
Right. So while the downstream machinery that actually builds male or female traits, genes like transformer, usually called tri and double sex or DSX, while that core part is highly conserved across most insects, the upstream signals, the very initial switch that says male or female, that part can be really novel and species specific. In bees, That novelty centers on a specific gene locus called CSD, which we need to talk about.
And just being a male bee, being haploid, must create some unique biological challenges, too.
Oh, definitely. Haplodiploidy has consequences. For male bees, making sperm involves a really heavily modified version of meiosis, that cell division that normally halves chromosome numbers. Their first meiotic division is actually abortive, it doesn't really work properly, and the second division is quite peculiar, leading to developing sperm cells of different sizes. And interestingly, several key genes that are essential for making sperm in flies are just missing in the bee genome. It highlights how specialized that process has become. Even something fundamental like dosage compensation, how males with only one set of chromosomes balance out their gene expression compared to females with two sets that's still not fully understood in male bees.
Wow. Okay, what about the germline itself, the cells that eventually become the eggs or sperm? This is another really striking difference from many model systems. In flies, for instance, the primordial germ cells, or PGCs, the precursors to eggs and sperm, they get set aside very early in development. They often bud off as distinct structures, like those pole cells we mentioned, or they're associated with specific material called the oosum. It's a clear early separation of the cells that will make the next generation from the cells forming the rest of the body, the somatic tissue.
But bees don't do it that way.
No. Honeybee embryos don't form those distinct pole cells or have a visible oosum structure early on. The PGCs only become clearly identifiable much later in development, located up in the dorsal, or back, part of the abdomen. Researchers tried using genetic markers that identify PGCs in other insects, genes like Visa and Nanos, to see if they could spot these cells separating early in bees. And while they found some brief early expression of these markers, there was no clear sign of a distinct group of cells segregating early.
So what's the conclusion? Either the germ cells form later, maybe induced by signals from surrounding cells, or they do separate early, but using completely different unknown genetic mechanisms that aren't shared with flies.
Exactly. It's another biological puzzle where bees seem to have found their own unique solution. It really forces us to think beyond the standard models.
Okay, let's dive into that locus you mentioned, CSD, the complementary sex determination locus. You said it's the key upstream switch for bee sex. How does that actually work, molecule by molecule?
Right. This is a fascinating story. It starts way back in the 1800s with a priest and beekeeper, Johann Ziron. He figured out just through observation that male bees must come from unfertilized eggs. Then, much later, in 1913, scientists actually looked at the chromosomes and confirmed males are haploid, females are diploid. That established the haplodiploidy basis.
But the real clue about CSD itself came from studying wasps, not bees initially.
That's right. In the 1930s, research on a parasitoid wasp, Brachyhepiter, uncovered something weird. They found diploid males. Usually males were haploid. This was a huge breakthrough because it showed that being male wasn't just about having one set of chromosomes. It had to be something about the specific versions or alleles of certain genes. Further work, particularly in another wasp, Habrobrachon juglandis, narrowed it down to one specific spot on a chromosome, the complementary sex determination locus, or CSD.
And what was the rule they figured out for CSD?
It's actually quite elegant. Females are heterozygous at the CSD locus. That means they inherited two different versions, two different alleles of the CSD gene, one from the queen, one from the drone she mated with. Haploid males are homozygous. They only have one copy, whichever single allele the queen gave them. And those rare diploid males, they are homozygous. They inherited two identical copies of the CSD allele. In honeybees, these diploid males are sterile, and the worker bees actually detect and eat them when they're tiny larvae.
Leaving those empty cells in the comb, that patchy brood pattern people sometimes see.
Exactly. That patchy brood became a visible genetic marker that researchers could use to track down the CSD gene itself. Two different research groups, one led by Greg Hunt and Rob Page, the other by Martin Bai, used genetic mapping. They basically walked along the chromosome, starting from the location linked to that patchy brood phenotype, until they found the gene. It turned out to encode a protein involved in RNA splicing, cutting and pasting RNA molecules, and importantly, it showed similarity to the transformer trade gene known from Drosophila. Then, using techniques like RNA interference or RNAi to basically shut down the CSD gene's function, they confirmed that the default developmental path in bees, without a functional CSD signal, is indeed male.
Okay, so heterozygosity at CSD is the trigger. How does having two different CSD alleles flip the switch for the rest of female development? What's the pathway?
Right, so based on what the sources tell us, here's how it's thought to work. CSD itself is the initial signal checker. If an individual is heterozygous, two different CSD alleles, this specific molecular combination somehow, through a mechanism that's actually still not fully understood biochemically, somehow leads to the production of a functional female-specific version or transcript of another protein called feminizer, or FEM.
And this feminizer protein is the B equivalent of the fly's transformer.
Yes, it's considered the B orthologue, the evolutionary counterpart of Drosophila trae. The FEM gene is actually located really close to CSD on the chromosome, probably resulting from an ancient gene duplication event. Once this functional female-specific FEM protein is made, it acts downstream of CSD. And interestingly, it actually promotes its own continued production, a positive feedback loop logging in the female pathway. This functional FEM protein then directs the processing of the RNA from the next gene in line, double sex, DSX, ensuring that the female-specific version of the DSX protein is produced.
Okay, and what if you're a male, either haploid or one of those diploid homozygous ones?
If you're homozygous, haploid male, or homozygous diploid male at CSD, you lack that critical heterozygous combination, so you don't get the functional CSD signal. Without that signal, the default happens. A non-functional male-specific transcript of FEM is made. And without the functional FEM protein, the DSX gene defaults to producing the male-specific isoform or version of the DSX protein.
So DSX is the major player further down, actually controlling the male versus female body parts.
Correct. DSX is a transcription factor. It controls whether other genes are turned on or off. In bees, the female version of DSX protein directs the development of female reproductive organs, like the ovaries and spermatheca. And studies using CRISPR gene editing to knock out DSX in females showed, yep, it messed up their reproductive development. Though interestingly, it didn't seem to affect sexually different traits in the head, suggesting other genes must control those.
So even though the very start of the pathway CSD heterozygosity is totally different from the fly pathway, which starts with counting X chromosomes and the sex-lethal gene, both pathways eventually converge on that same core control mechanism involving TRE, or FEM in bees, and DX?
Exactly. That TRADSETS axis seems to be a deeply conserved module across most insects, responsible for executing the male or female developmental program. It perfectly illustrates that mosaic concept. Again, you have unique, novel upstream trigger in bees, CSD, plugging into conserved downstream machinery, FEMSEX. It really underscores why we need to study diverse organisms to see both the common threads and the unique biological inventions.
Right. So from setting that male-female path in the tiny egg, let's move into that period of absolutely explosive growth, the larval stage. What's life like for a honeybee larva?
Well, for a larva, life is pretty much about one thing: eating and growing incredibly fast. We're talking about huge increases in mass compared to when they hatched. A worker larva gets about 500 times heavier. A queen larva, around 1,600 times, and a drone larva, a staggering 2,300 times heavier, all in just 2 or 3 weeks.
That's just mind-boggling growth. And they let the nurses know they're hungry.
Yeah, they produce a brood pheromone. It's a chemical signal that basically says, feed me, to the nurse bees.
Yeah.
All this rapid growth means they constantly outgrow their skin, their exoskeleton. So they have to molt shed the old, tight exoskeleton, kind of like wriggling out of a suit of armor that's too small, revealing a new, larger one underneath.
How many times do they have to do that? Shed their skin?
Honeybees go through 6 larval instars. An instar is just the stage between molts, and that number 6 is the same for all 3 castes: workers, queens, and drones.
Okay, can you walk us through those larval stages quickly?
Sure. The first 4 instars are pretty short, about a day each. The larva is curled up in a C shape in its cell, which is still open, and it's just constantly being fed by nurse bees. The 5th instar is longer. For the first couple of days, it's still being fed and grows really rapidly. Then the nurses provision the cell with a final larger amount of food, and they cap the cell with wax.
Ah, capping the cell. That must signal a major transition.
It does. Once capped, the larva enters what's often called the spinning stage. This lasts about 37 hours or so. It straightens out inside the cell and spins a silk cocoon around itself, lining the cell walls. It uses secretions from its salivary glands to this. This stage is also when its gut development finishes, and it has its one and only defecation, clearing out waste before the next big change.
And then it sheds that larval skin one last time.
Yes. That final larval molt is called apolysis. The old larval cuticle separates, and underneath, the new pupal cuticle has already formed. This stage, between the larval molt and becoming a fully formed pupa, is often called the propupa. And crucially, during this prepupal stage, something really important starts happening internally. The imaginal discs begin their major development.
Imaginal discs. Those are the little packets of cells set aside way back in the embryo.
Exactly. They've been sitting dormant, and now they start rapidly growing and differentiating. These discs are destined to become the adult structures: the legs, wings, antenna, eyes, mouth parts, gonads, everything.
So even while it still looks like a larva from the outside, the prey pupa is already starting to build the adult bee inside.
Pretty much, yes. The groundwork for the adult form is being laid down internally.
And that leads us directly into the pupal stage, the real heart of metamorphosis.
The pupa is technically the sixth and final instar, and this is where the most dramatic transformation occurs. It's a massive internal reorganization, and honestly, the details are still not fully understood, especially in bees compared to flies. Many of the larval organs, like the specialized gut and the fat body, are basically broken down, collateralized, and their components are recycled to build the adult structures from those rapidly developing imaginal discs. The nervous system also gets a major overhaul. Certain brain regions grow, and nerve clusters, called ganglia, along the ventral nerve cord fuse together.
What about changes we can actually see on the outside during pupation?
Well, when it first molts into a pupa, it's white and very soft, kind of like a ghost of the bee it will become. Gradually, it starts to gain color. The eyes usually darken first, turning from white to pink to purple to black. Underneath that pupal skin, the new adult cuticle is forming. The stage when the developing adult is inside the pupal cuticle is called the ferret adult. As development finishes, the adult cuticle pigments fully and hardens or sclerozes.
And then finally, emergence.
Right. The bee sheds that final pupal cuticle, chews its way through the wax capping of its cell, and emerges as what we call a teneral adult. At this point, it's still quite vulnerable. Its cuticle is soft, it's often lighter in color, quite fuzzy, and it can't fly properly or sting effectively yet. It needs a bit of time for its exoskeleton to fully harden and for some final maturation before it can start its adult duties in the hive.
So we've traced the journey. Simple egg, rapid larval growth, incredible pupal transformation, and the newly emerged adult. But we skipped over that huge decision point for female eggs, becoming a queen or a worker. That's caste determination, right?
Yes, exactly. Caste determination in honeybees is just a fantastic example of polyphenism. That's the phenomenon where you have individuals with the exact same genes, the same genetic blueprint, but they develop into drastically different forms, different phenotypes, purely because of environmental cues they experience. For diploid female bees, that environmental cue, experienced during the larval stage, dictates whether they become a fertile, long-lived queen or a typically sterile, much shorter-lived worker. It's a central puzzle in insect biology because it tells us so much about how genes and environment talk to each other.
What are the big questions researchers are trying to answer about this queen-worker switch?
There are basically three main levels. First, what exactly is the environmental trigger? What signal in the larva's world says become queen or become worker? Second, how does that external signal get translated inside the larva? What internal pathways, especially hormone pathways, the endocrine system get activated differently? And third, how do those internal signals ultimately control the massive differences in gene expression and development that lead to the distinct queen and worker bodies and behaviors?
Okay, let's start with the environmental trigger. It's all about the food, isn't it? That's the classic story.
Differential feeding, yes. That's been known for a very long time to be the main environmental signal. Nurse bees produce this special brood food in glands in their heads, the hypopharyngeal and mandibular glands. Now, for the first 3 days or so, all young female larvae get basically the same mix. But after that, their diets diverge dramatically. Larvae destined to be queens are fed exclusively royal jelly. This is a rich food, slightly favoring mandibular gland secretions, and importantly, they get it throughout their entire larval development. And crucially, they get loads of it. They're practically swimming in royal jelly in those big queen cells. Worker larvae, on the other hand, after those first 3 days, they get switched to worker jelly, which has more hypopharyngeal gland secretions. And around the third instar, their diet also starts getting supplemented with pollen and honey. Plus, they only get fed just enough to grow no huge surplus like the queens.
So it's both the type of jelly and the amount they get fed.
Both seem to be really important, and untangling them is tricky. One key difference between royal jelly and worker jelly is the sugar content. Royal jelly has significantly more sugar. Early studies showed that if you took worker larvae and fed them worker jelly but just added extra sugar, you could nudge them towards developing queen-like traits. This led to the idea that maybe the high sugar in royal jelly acts as a feeding stimulant, just making queen larvae eat more and grow faster. However, more recent work is suggesting that the sheer quantity of food might be even more critical than the exact composition. Since worker larvae are naturally restricted in how much they get, it's hard to fully separate the effects of, say, specific proteins in royal jelly versus just the effect of having unlimited food available.
Are there other things in the food besides basic nutrition and sugar that might be acting as signals, like specific molecules?
Researchers are definitely looking into that. For instance, worker jelly seems to have higher levels of certain microRNAs. These are tiny RNA molecules that don't code for proteins but can regulate other genes. Experiments where they added worker jelly microRNAs to royal jelly fed to larvae showed a bias towards worker development. So microRNAs might be another layer of control, maybe affecting things like brain development. Another molecule getting attention is 10-HDA. It's a fatty acid found in higher amounts in royal jelly. What's interesting is that 10-HDA is known to inhibit enzymes called histone deacetylases.
Enzymes that affect how DNA is packaged, something related to epigenetic.
Exactly. So it's a potential direct link between diet and epigenetic changes. Experiments have shown 10-HDA can affect body size and gene expression in bees, but its precise role in causing caste determination is still considered, well, a bit unclear. It's preliminary work.
So thinking about this trigger, is it like a simple light switch? Food A, worker, food B contakes queen, or is it more complex?
That's a great question. In many insects with polyphenism, it is like a simple switch. You get one trigger, and it flips development cleanly into one of two distinct forms, with nothing in between. But in honeybees, we see something that complicates that picture: the existence of intercastes. These are individual bees that show a mixture of queen traits and worker traits.
Intercastes, like a bee with partially developed ovaries, but maybe a worker-sized body?
Exactly. They have characteristics of both.
Yeah.
If caste determination was just a simple on-off switch, you wouldn't expect to see these intermediate forms. You'd get either a perfect queen or a perfect worker. The fact that intercastes exist suggests it's probably a more complex, maybe even a continuous developmental process, one that can be pushed towards the queen end or the worker end, possibly with different factors influencing different traits at different times. And what's really revealing is that when scientists rear larvae in the lab, say on royal jelly plus extra sugar, they don't just get queens and workers. They often get a whole range of intermediate forms, almost like a Biddle curve distribution.
Wow, so that could mean intercastes are almost the default state, and it's the very specific intensive feeding by nurse bees that pushes development all the way to the extreme queen or worker phenotypes.
That's definitely one interpretation supported by the data. It also highlights how flexible the system is, at least for a while. You can actually induce queen-like traits in older worker larvae, even up to the end of the fourth instar, just by switching their diet to a queen-like one. Differentiation isn't rigidly fixed early on. It seems to be an ongoing process right up until the cell is capped.
Okay, so the differential feeding is the complex external cue. How does that signal get transmitted inside the larva to actually change development? What are the internal, hormone pathways involved?
Two major signaling pathways are consistently implicated here. First, there's the Eister pathway. That stands for insulin-like signaling and target of rapamycin. These are fundamental pathways found across animals, deeply involved in sensing nutrients and controlling growth. The other key players are the classic insect hormones, juvenile hormone, or JH, and the echetosteroids, which control molting and metamorphosis.
And these pathways behave differently in larvae developing into queens versus workers.
Oh, significantly differently. For example, the levels of juvenile hormone, JH, are much, much higher in queen larvae during the later stages compared to worker larvae. And crucially, if you experimentally treat worker larvae with extra JH, you can trigger queen-like development. JH also seems to prevent the programmed cell death, the apoptosis of the developing egg tubes, ovules, in the ovaries. This is key because that cell death is what makes worker ovaries non-functional. JH seems to be acting downstream of those Eister nutrient-sensing pathways. Ecdystroid levels also show distinct patterns between castes, and there are even different versions or splice variants of the ecdystroid receptor protein. The interplay between JH and ecdystroids is complicated and actually changes during development. Their caste-specific levels affect ovary development, cuticle formation, even brain structure.
Right. And what about the Eister pathway itself?
Well, the gene for TOR itself is expressed at higher levels in queen larvae. And experiments using RNA interference, that technique to reduce a gene's activity, showed that knocking down either 2R or the insulin receptor gene biased development toward worker phenotypes, even if the larvae were fed a queen-promoting diet. So Eister seemed to be acting upstream, likely sensing the nutrient differences in the diet and translating that into signals that influence growth rate, body size, and probably also downstream hormone levels like JH.
But you mentioned Eyster are fundamental to growth in general. Isn't it hard to be sure that knocking them down is specifically affecting caste rather than just messing up growth overall?
That is a really important caveat. Because Eyster are so central to basic metabolism and growth in pretty much all animals, crude knockdown experiments can definitely have general negative effects on development and size, regardless of caste. It makes it tricky to definitively say this effect is purely about the caste switch versus this is just because we've stunted the larva's growth. That's why researchers are increasingly turning to more precise tools like CRISPR gene editing, hoping to target specific components of these pathways more cleanly.
Okay, so beyond hormones and nutrient signaling, there's this other layer, epigenetics. How does that fit into the queen worker puzzle?
Epigenetics is fascinating. It refers to changes in how genes are used, which ones are turned on or off, that can be inherited when cells divide. But importantly, these changes don't involve altering the actual DNA sequence itself. It's all about gene regulation, controlling access to the DNA blueprint. The two main mechanisms people talk about are DNA methylation and histone modifications.
Can you break those down quickly?
Sure. DNA methylation is like putting tiny chemical sticky notes, methyl groups, onto the DNA molecule itself. In bees and other insects, this often happens within the main coding part of genes, not just at the on-off switch like in mammals. It seems to be involved in regulating everyday housekeeping genes. Histone modifications are chemical tags added to the histone proteins, the spools the DNA wraps around. These tags change how tightly or loosely the DNA is packed. Tightly packed DNA is harder for the cell's machinery to read, so those genes tend to be off. Loosely packed DNA is more accessible, so those genes can be turned on. Together, these influence chromatin structure and gene accessibility.
And what have scientists found about epigenetics and bee castes?
Well, remember that fatty acid 10-HDA in real jelly. We mentioned it inhibits histone deacetylases. That's a direct potential link from diet to histone modifications and changing chromatin structure. More direct evidence came from experiments targeting DNA methylation. Researchers used RNAi to knock down the main enzyme that adds methyl groups, called DNMT3. Reducing DNMT3 activity strongly biased larval development towards the queen phenotype. The effects were especially dramatic on ovary development, allowing them to become functional like a queen's. We also know that the patterns of DNA methylation are different in the brains of adult workers and queens. And very recently, studies have looked closely at histone modifications during larval development. They found lots of these modifications, and crucially, specific types of marks showed different patterns in queen versus worker larvae. These patterns correlate really well with which genes are turned on or off differently between the castes. Some histone marks seem to act like enhancers, boosting the activity of genes needed for worker traits.
So epigenetics seems like another crucial layer of control, maybe acting as the memory system that translates those early dietary signals into the long-lasting patterns of gene expression needed to build and maintain a queen or a worker.
That's definitely the picture that's emerging. It's a really hot area of research right now. And interestingly, because fruit flies actually lack some of the key DNA methylation machinery that bees have, the honeybee is really well positioned to become an important model system for studying how epigenetics works in development more broadly.
This has been a truly incredible deep dive. We've followed the story right from that seemingly simple egg, through its explosive larval growth, the amazing pupal transformation, to the final adult bee. And critically, exploring how its sex gets determined, and for females, how that environmental nudge sends them down the path to being a queen or a worker.
It really highlights this amazing interplay, doesn't it, between the basic genetic instructions, the signals coming from the environment, especially the larval diet, then the cascade of internal hormone signals, and now, overlaying all of that, these intricate epigenetic controls.
And it absolutely drives home the value of studying organisms like the honeybee, things that aren't our standard lab models. They show us that while some biological processes are deeply conserved across life, evolution has also produced completely novel, sometimes startlingly elegant solutions to fundamental developmental challenges. Solutions we just never see if we only looked at flies or mice.
Exactly. They constantly challenge our assumptions and force us to broaden our understanding of how life works.
So, as we finish up this exploration, here's something to think about. How is it even possible that the exact same set of genes sitting inside one female honeybee egg can result in such radically different creatures, a queen built for reproduction and longevity, or a worker built for labor and self-sacrifice? A fate determined largely not by their genes, but by the food they eat as a larva, setting off waves of hormones and epigenetic marks that shape them for life. What does this incredible developmental flexibility, this profound power of the environment to sculpt biology, tell us about the potential influence of our own surroundings, our own experiences on shaping who we become? Something to chew on until our next deep dive.